am Sophia, and today I'm going to talk about the polymerase chain reaction. The polymerase chain reaction makes copies of our DNA, has many real-life applications, and detects sequence variations in specific cells or organisms. First of all, what is DNA? DNA stands for deoxy, so it lacks an oxygen. More specifically, it lacks a hydroxyl group. So this S on the sugar phosphate backbone. In ribose, it has two hydroxyl groups. In deoxyribose, however, it has only one. Ribo again means it's from the sugar ribose, and nucleic means it's normally in the nucleus, but DNA can be transferred to other parts of the cell. And it can, for example, be in the mitochondria, mitochondrial DNA, totally possible, and even chloroplast DNA. And it can be transferred between cells. But normally it's in the nucleus. DNA is composed of 4 nucleotides. Adenine A, that's the orange nucleotide. Thymine T, that's the green nucleotide. Cytosine C, that's the gray nucleotide. And guanine G, that's the purple nucleotide. Adenine pairs with thymine and cytosine pairs with guanine. And complementary nucleotides are nucleotides that pair up together. And they pair up together through hydrogen bonds. You can see the A has these 2 dashed lines to the T, and the C has those 2 dashed lines to the D. Those dashed lines are the hydrogen bonds. A DNTP is a nucleotide with 3 phosphates. And a DDNTP, or a dideoxynucleotide, is a DNTP without any hydroxyl groups except the hydroxyl groups on the phosphates. So you can see down there, there's this HO and OHs attached to the P's, phosphoruses. Those are the hydroxyl groups on the phosphates. So now, what is the polymerase chain reaction? The polymerase chain reaction, PCR, is a reaction used to make millions or billions of copies of a sample of DNA in only hours. The DNA is copied in a PCR tube instead of in an organism so that the DNA can be analyzed. And in PCR, the DNA is cycled many times through a series of heating and cooling changes. And these heating and cooling changes are automated through a thermal cycler, which I'll talk about later. So you can see right there on the image, there are those 8 PCR tubes that were just removed from a reaction, from a PCR reaction inside a thermal cycler. As you can see, they contain amplified DNA, mainly amplified DNA. So what are the ingredients for PCR? PCR requires the following ingredients. First, the DNA template, which contains the sequence to amplify. If you have no template, then you can't replicate it. Then, a DNA polymerase that adds complementary nucleotides to the template. You can see on the image, there's this blue DNA polymerase that is connecting the free nucleotides with the already existing DNA template strands. But the DNA polymerase needs a little help. It needs 2 primers. Those primers are responsible for providing a small sequence of DNA, which is necessary for the polymerase to add nucleotides. Then there's the DNTPs, the deoxynucleotides, which are the building blocks for the polymerase that it needs to synthesize a new strand, and a buffer that provides a chemically suitable environment for the polymerase. And lastly, MgCl2, magnesium chloride, which is needed for the enzyme to work, more specifically the polymerase enzyme. So what are the steps of PCR? The first step in PCR is denaturation. In denaturation, the ingredients are heated to 94 to 98 Celsius for 20 to 30 seconds. The hydrogen bonds between the strands of DNA are broken and two single-stranded DNA molecules are formed. So you can see right next to the X, there's this double helix. And this double helix is normally how DNA is made. So DNA is normally in this double helix form. But you can't use that double helix to copy DNA through PCR. So first, you need to have an initial denaturation. This initial denaturation unwinds the double helix. Then after that, there'll be another step of denaturation that is conducted every single time. And that is when you already have one long strand. So you have one long strand and another long strand, but they're connected together. And they need to be broken apart before you can continue, because then you're going to add other things to it. In annealing, the second step of PCR, the ingredients are cooled to 50 to 65 Celsius for 20 to 40 seconds. So in annealing, you're adding this pink and yellow primers to these disconnected long strands. So now that you have the disconnected long strands, you can add the primers, which are in turn helping the polymerase enzyme. It is critical that the correct annealing temperature is determined prior to PCR because it affects efficiency and specificity. Because there might be issues with primer binding, which is very important for future steps in PCR and for the next cycle. The temperature for extension, the third step of PCR, varies depending on the polymerase. So once you have extension, but the problem is, what polymerase are you using? That's a big question. But that doesn't have one right answer. But it has one answer that is used, one of them is used very commonly, but the other ones are used in certain cases where it's very important to have high accuracy or efficiency or other items that are missing from the polymerase that we commonly use. So the polymerase that we commonly use is Taq polymerase, and its temperature is 75 to 80 Celsius, usually 72 Celsius in extension. So that Taq polymerase, after that, but there is one problem with Taq polymerase. The problem is that Taq polymerase has no proofreading system. In DNA replication, copying of DNA needs to be almost perfect because DNA replication comes right before cell division. So you need to have this DNA properly copied. The polymerase is very robust and adds nucleotides almost always correctly, but it has some errors. And those errors would happen way too often when a cell would divide, and that would lead to issues such as mutations, which could cause disease or other things that are harmful. So these mutations need to be corrected before they happen. So there's this proofreading system, and the proofreading system is very robust and increases the efficiency to around 1 in a billion. So 1 in a billion base pairs is added erroneously, and that's much better, especially if there's 6 billion base pairs in a genome. So you can see that. But the issue with Taq polymerase has no proofreading system. If Taq polymerase has no proofreading system, remember, PCR is normally used in mutations, in mutagenesis, mutation research. It's normally used to check for mutations, but to check for mutations, you need to have a high efficiency polymerase enzyme. And in that case, you're going to use a PCR enzyme, polymerase enzyme, that has a proofreading system. But what about PFU polymerase? PFU polymerase does have a proofreading system, and it's better than no proofreading system at all. But PFU polymerase's proofreading system is not as robust as the proofreading system in our DNA replication polymerases. So we use high-fidelity DNA polymerases. Even these polymerases make errors, but those errors happen much less often, and that makes these high-fidelity DNA polymerases suitable in mutagenesis and other research dealing with mutations. So in extension, complementary nucleotides are added to the copies of the DNA template. Annealing temperature and primer binding. So in annealing, the primers must attach to the DNA perfectly, or almost perfectly. Nothing can be perfect. But if the temperature is too low, then the primer might bond imperfectly. But if the temperature might be like just a little off, if the temperature's a little off, then maybe the results will be the same or very similar in terms of efficiency and other important characteristics. But if the temperature is a little lower, then there might be some issues. There might be a reduced efficiency and other important characteristics which are due to the primer misbinding, and no polymerase enzyme can fix any of those problems. So if the temperature is too high, then the primer may not bond at all, or bond imperfectly again. So how can this be fixed? This can be fixed using so-called high-fidelity DNA polymerases, and these high-fidelity DNA not high-fidelity DNA polymerases, but it can be fixed using so-called PCR master mixes. Not all of them, but some of them offer what is called a universal annealing feature, which has one annealing temperature for PCR reactions, and that's helpful. It offers another advantage, conducting multiple PCR reactions at once, because usually they have different annealing temperatures too. PCR in real life. What are the real life examples? PCR permits rare species to be identified, so PCR is commonly used when searching for pathogens such as coliforms in water supplies. PCR-based technologies are used every day in clinical diagnostics, forensic investigations, and agricultural technology. PCR can be used to detect sequence variations in specific cells or organisms, for example, mutations in humans or home pets. A water supply is the provision of water by public utilities, commercial organizations, community endeavors, or individuals. Water from water supplies usually passes through filters. These filters prevent pathogens in the water from infecting people. These filters are not 100% efficient, and some pathogens may bypass some or all filters. Let's say a pathogen in a pipeline from a neighboring pipeline enters another pipeline, and it has bypassed the filters in the neighboring pipeline, or maybe the neighboring pipeline has no filters. But there's 2 filters left over in that pipeline. So maybe most of it gets filtered, but maybe this particular pathogen doesn't get filtered. And it bypasses the filters, all of them. And then it comes into whoever gets water from that pipeline. Pathogens are infectious agents or germs. These pathogens could bypass water filters and impact water quality. An example for waterborne disease is hepatitis A virus. First, a sample of water is amplified via PCR. Then a clean sample of water is also amplified. And then both samples are then sequenced and compared to detect changes. So you can see first, the second image shows very, very dirty water. So here, this very dirty water gets amplified via PCR. Then this very clean water gets amplified via PCR too. And then the samples are sequenced and compared. So you can see an open Chrome. You can see in the miscellaneous tab, you see a random string of letters, but it's composed of the same nucleotides that DNA is composed out of. A, T, D, and C. So it's a DNA sequence. Here you see a very long DNA sequence. But most likely there'll be mutations along the way. Maybe they're big. So the mutations, you could see a mutation like CAA-GATT is the control, and CA-GATT is the mutated sequence. Oh, this is a deletion mutation. This is very harmful. Deletion mutations are harmful. Very harmful. Almost all of them are harmful. And you can see the insertion mutations. Maybe you could get CAA-GATT, so 3 A's instead of 2. No, that's not good. That's going to be an insertion mutation. Most of those are harmful too. And then you've got a mutation like CAT-GATT. Oh, that's the A. 1A got replaced to AT, and that's a substitution mutation. While most of them are harmful, some of them could be neutral or helpful. But most of the time they're harmful too, but might require not laboratory analysis. Agriculture is the science, art, and practice of cultivating plants and livestock. But certain plants may get infected by a pathogen and die. After a few days, most or all of the plants may die. If all of the plants die, then farmers might need to wait for a year for new crops to grow, or maybe even longer. Pathogens can also infect plants and spread rapidly. They're so small that they cannot be seen using a light microscope. And even if they would be seen using a light microscope, in certain cases, some exceptions, then a light microscope is usually not present. It's usually not used on a farm. So what's the use of PCR in agriculture? First, a sample from an infected plant is amplified. So you can see an infected plant. And then a sample from a healthy plant is amplified. For example, a plant from another field. And they're also sequenced and compared using a program like OpenChrome, but this time they're compared to check if they're the same or very similar in certain regions. If they're the same or similar, then the plant contains a pathogen. And if the plant contains a pathogen, the plant must be infected. Forensic applications. PCR is used in forensics to find the suspect at a crime scene. PCR is necessary because usually the fragments are very small. The DNA samples from the crime scene and the samples from the suspects, are amplified. Then the fragments are electrophoresized using a DNA ladder. If one of the suspect's bands matches the crime scene's bands, then that suspect committed the crime. So in this image, you can see this first line is a marker. But you can compare the crime scene to them. So that if the crime scene would have 1,000 base pairs, 1,000 base pair marks, then it could be either A, B, or C. That doesn't make sense. But maybe that could eliminate something. Like if it has a 900 base pair mark in addition, then it must be sample A. If it is an 800 base pair mark, however, it must be sample B. But maybe it's not that straightforward. Maybe it has some mark that is not present in any of the 3 marks. But then you just take your best guess. Maybe you just missed some samples. Maybe you just... So you could see maybe it has 600 base pair mark, then it must be sample C. But if it would, for example, have like around 300 base pair mark, then no, that would be harder to determine because it's in 2 samples. But then if it also has 100 base pair mark, then it must be sample B. So you can determine that, but it's not always as straightforward. Maybe it also has a 400 base pair mark, and you can see the 400 base pair mark is only on the marker. And if none, so in this case, none of A, B, or C have that 400 base pair mark, then oh no, then you can figure out, oh, neither suspect A, neither B, neither C conducted this crime. Maybe we're searching for the wrong crime, or maybe we need to take more suspects. Maybe we need to have a D, E, and F and compare those and see if they have a 400 base pair mark. So sometimes it doesn't just like match, but you can figure out what to do based on when it doesn't match. You might see maybe it matches quite closely. If it matches quite closely but not exactly, you might infer that suspect committed the crime. Well, if they completely don't have a mark or anything close to it, then when you figure out, oh, neither of these suspects committed the crime. So maybe you're searching the wrong crime. Maybe you're just using the wrong suspects. So you can just figure out what's happening. So on the bottom, you can see 2 of these samples match. So you can see the 4th and the last sample match. So they're almost exactly or almost exactly in line with their DNA markers. Matching doesn't mean perfect matching. It doesn't mean perfect matching. But it means that either perfect or almost. They're like almost. So gel electrophoresis. We use gel electrophoresis in forensics. So once the DNA template is amplified, you can figure out the approximate length of the sample by gel electrophoresis. Gel electrophoresis separates fragments based on how far they are from the positive end. So let's say on the very edge, on the second image, there's a mark of 2,000 base pairs. And let's assume that all the marks are stripped away from this ladder except the 2,250 base pair mark. In this case, there's a DNA sample that's in the middle. It doesn't hit any of these marks, and it's not even close to them. Then you'll be able to find out what the approximate length is. It will be between 251 and 1,999 base pairs. But let's say we include the 1,500 base pair mark, and it doesn't hit it. If it does hit it, then ta-da, we've got the length of the sample, but in most cases, you won't exactly hit, or you won't even get close to the second mark. So 1,500 base pairs, you just change it. Maybe you get something that's far away from 1,500 base pairs, so you can see, oh, it's somewhere between 1,499 base pairs and 251 base pairs. And maybe there's 1,000 base pair mark. And it doesn't quite hit that, and it isn't even close. So you can say, oh, it's somewhere between 999 and 251 and 999 base pairs. But let's say there's a 500 base pair. There's a 750 base pair mark. if there's that base pair mark, then you can say, oh, it doesn't hit that yet. So if it does hit the 750 base pair mark, oh yeah, the whole thing hits the 750 base pair mark, then the whole thing is 750 base pairs long. But in most cases, that's not the thing. There's going to be multiple fragments. Let's say one of them is at 750. But let's say we figured out the approximate length of another DNA sample, and it hits around 500 base pairs. That would be 2 marks. So one segment of the DNA is 750 base pairs long. The other segment of the DNA is 500 base pairs long. So why is it less? So in the first place, we had compared the length of all these DNA samples. We compared the length, but why does that happen? So why is it less than 2,000 base pairs if it doesn't hit it? Well, because it's further away from the negative end, or further away from the positive end than 2,000 base pairs. And if there would only be the 250 and 2,000 base pair mark, which is almost always not true, then there would be, then it would be closer to the positive end than the 250 base pair mark. The fragments produce a pattern of lines that can be compared to the crime scene. So up to 58% of human DNA is repetitive. 2 categories of repetitive DNA, short tandem repeats and variable number tandem repeats, can be used to identify criminal suspects. So you can see VNTRs, the variable number tandem repeats, are usually 10 to 100 base pairs long, and short tandem repeats are usually 2 to 10 base pairs long. So the short tandem repeats and the variable number tandem repeats are repetitive DNA, and there's 2 categories, big categories of repetitive DNA. Repetitive DNA that includes transposable elements and related sequences, which is 44%, and repetitive DNA unrelated to transposable elements, which is 14%. So PCR can amplify VNTRs and STRs with primers that flank each of the repetitive regions. The sizes of the fragments from any individual for each of the STRs will show what alleles are present. When several STRs are analyzed for an individual, the size of the STR fragments will reveal a set of likely unique alleles. So you can see here in the image that the VRC1 was sequenced and it matches, VRC2, VRA, VRB1, VRB1, VRB2, they match, all of them match, but there might be some other alleles. This is a very rare case, but you can see that right next to, like, one band away from the red band that is in the middle of VRC2 and VRA, one of the alleles is present and another is absent. It's very hard to notice. Some of them are slightly brighter or dimmer, but they're super hard to notice. This is a very rare scenario. Usually there would be one gene that would be present and another absent, or there would be one gene that is significantly brighter on the electrophoresis diagram than the other gene. So the restriction enzymes. The restriction enzymes are enzymes that recognize and cut DNA at a specific palindromic base sequence. PCR can be used to amplify the DNA, which can increase the chance that a fragment of the gene is found. PCR products must usually be manipulated and cleaved using restriction enzymes. And if there's no recognition site, then the PCR product may only be partially or not digested. So you can see on the image that it's usually on the very end of the recognition site, the restriction enzyme. And then there's this restriction enzyme, BAMHI, which cuts up the sequence at a specified base sequence. And then it usually leads either sticky ends or blunt ends. So in this case, the enzyme makes two sticky ends that can be reconnected to each other. A very current use of PCR is SRS-CoV-2 testing. So PCR is the gold standard for this testing since it detects genetic material specific to the virus and is more sensitive than antigen tests. And the test can be done anywhere, such as in a clinic, hospital, or car. So you can see this person that is conducting a PCR test for the virus there. So you can see that here the results are usually going to be available within around 1 or 2 days, but maybe longer. So yeah, it's better than antigen tests because research states that antigen test sufficiency is around 80% when the person is symptomatic for the virus. But it's only around 40% efficient when the person is asymptomatic, which when you get negative antigen tests, then you most likely will need a PCR test for a confirmation, just in case, to see that you really don't have the virus, especially if you're asymptomatic. So detecting sequence variations. Someone was in close contact with someone else carrying virus X. Viruses like virus X can conduct mutations. They can mutate DNA. Sickle cell anemia can also do the same thing. It is a disease that can make mutations. So in certain cases, a virus could mutate the DNA. Once a virus enters the cell, it can just hijack the cell machinery, and normally it would replicate its own DNA, but its own DNA in certain cases could mutate certain spots of the already existing host cell DNA. But most of the time, this doesn't happen with viruses. Most of the time, mutation arise just by errors in DNA replication. There's no way you can predict these issues. But viruses like virus X might have a delay between acquiring the virus and infection, and it could have a delay between infection and first symptoms. And mutations might have similar delays. So maybe suddenly there's a strand of DNA that got replicated with a few errors. And let's say most of these errors were corrected by the proofreading system, except one error that changed a C to a T in a certain base pair. It's a very hard to notice mutation. But what does that do? Let's say the change from a C to a T makes a big mutation that causes virus X or something similar. So you can see with that, maybe a week or something passes, and you just literally don't notice you have a mutation. There is a delay between acquiring of the mutation and when you get your first symptoms. And in certain cases, mutations can have no symptoms at all, or very hard to notice symptoms, such as in sickle cell anemia. It's hard to notice unless you have low oxygen. So, But after that delay, you could maybe notice, oh, I have this disease. But maybe, in certain cases, it can even be an unknown mutation that isn't even tied to a disease. Maybe the symptoms that you experience never happened with this given disease before. And you need to go to the hospital right away, but you don't even know what disease you have. So PCR can help you fix this problem of, hey, I have this mutation, but like it's super rare or nobody else had these symptoms. So it isn't like linked to a specific disease where you can figure out, oh, this is a given disease and this is this disease and this mutation makes this disease and this other mutation is this disease. You can't, it isn't in any list of known diseases. Oops. So what are the requirements? First, you need your DNA sample, a primer, so 2 primers, and a thermal cycler to conduct the PCR. So the sample and primers are inserted into a PCR master mix, and then the PCR master mix is inserted into the thermal cycler. But there's a few issues. You want to stay for a little. First, you have to take an empty PCR tube from the bottom of the thermal cycler, or if you use a PCR master mix, like the one on the second picture, you should take an empty tube of that PCR master mix. So you'll need to place the DNA sample and primers into the PCR master mix and place that into the tube and the tube into a blank space in the cycler. So you can see the bottom of a thermal cycler. It can be placed in any of these holes. And this right here is the PCR master mix. The second step is to adjust the temperatures and weight. Now I have to adjust the denaturation temperature to around 94 to 98 Celsius, the annealing temperature to 60 Celsius, which is very important because the PCR master mix has a universal annealing temperature, and the extension temperature to 72 Celsius. Then wait for up to 4 hours. So you can see you have these blocks and you must set them to the correct temperature, and maybe you have multiple PCR reactions running. They might be assigned under different temperatures, so make sure you assign the correct block with the correct temperature. That was on the biometric. Try a thermal cycler on the Eppendorf thermal cycler. You can see, make sure your temperature, like cycle 1, if you see that the temperature is incorrect, so it's not 60.0, then oh no, there might be an issue. You might have a little of a problem here. And it's better to figure it out and check the temperature for the first cycler. It's good to check the temperature just in case you get an issue. So you better to stop, abort, or exit than to just waste your time using the wrong temperature. And maybe mutations are more common or the efficiency is bad or something like that. That usually happens with issues with the annealing temperature. So once your DNA template is amplified, you can detect large insertion and deletion mutations by gel electrophoresis. Gel electrophoresis requires a gel electrophoresis apparatus, agarose gel, and PCR tubes with the amplified DNA samples. Then place the DNA ladder and insert the DNA sample into the electrophoresis apparatus. And then pour in all the agarose from the agarose tube and wait 15 minutes. So you can see you should insert it at this blue arrow, the insertion point. That's where you're going to insert all the items. The agarose gel should just be spread out everywhere. If one of your DNA samples was from a red blood cell, then if you see any purple instead of black bands, then you have malaria. So the results of gel electrophoresis. The first lane is the marker that shows all the lanes on the DNA ladder. The second lane reveals the approximate base pair length of the control fragment. And the third lane represents the approximate base pair length of the possibly mutated fragment. So you can see the first is just a marker. The second one is this 250 base pair mark of intensity, so that's 2. And then 3 is another 250 base pair mark, so they match. They match. Might be a photographic error that it goes a little on the top. But you can see, whoa, it already matches. They match perfectly. So you can see that the 250 base pairs match perfectly. You've got them. So there is no mutation in this individual. So why is DNA sequencing necessary? Gel electrophoresis can identify whether a mutation is an insertion or deletion mutation, which are harmful. Mutations can also be substitution or inversion mutations. DNA sequencing is used to find out whether a mutation is a substitution mutation. And if you analyze DNA sequencing results, then you'll get data of how frequently a base appears, and you might get something like fluorescent dyes that display the exact sequence of the DNA you had. And this is extremely helpful. So DNA sequencing. You can find any other mutation by DNA sequencing. DNA sequencing reveals the sequence of base pairs of any DNA fragment. And a DNA sequencer automates the process of DNA sequencing. DNA sequencing requires amplified DNA from the PCR tube unless it's next generation or third generation sequencing that you're conducting. You're conducting next generation or third generation sequencing, which this sequencing machine does not use. It uses the regular sequencing. But depending on the method, sequencing may require electrophoresis if it's next generation or third generation sequencing, ddNTPs, or 4 fluorescent dyes in different colors. So you can see the 4 fluorescent dyes in different colors, which will just be nicer, and it will show you what the sequence is. So be much more recognizable what base pair is what, and if it's much more recognizable, then it will be easier to find errors like mutations. It will just be a little more convenient to do so. So if a mutation is an insertion or deletion mutation, you need to go to the hospital. Some insertion mutations are large segment duplications, which are neutral since they only change non-coding DNA. Most mutations that change non-coding DNA, except for the mutations that change regulatory DNA, are neutral. Most insertion mutations insert multiple bases and change coding DNA. But almost all deletion mutations delete either coding DNA, which is harmful, or regulatory switches, which is even more harmful because it might affect a different chromosome, a different cell, a different tissue, or even a different organ system. If a mutation is a substitution mutation, then the mutation will require laboratory analysis. Most substitution mutations are harmful, but some substitution mutations are neutral or helpful. Substitution mutations are more often neutral or helpful than other mutations. But otherwise, the mutation is an inversion mutation, and it also requires laboratory analysis. Inversion mutations are very hard to detect since they deal with multiple chromosomes. At the minimum, 2 random chromosomes from the cell need to be both fully sequenced, but normally the entire cell needs to be sequenced. If we're dealing with regulatory sequences, however, the entire genome may need to be sequenced, which is around $1,500 even with third-generation sequencing. PCR can replicate minuscule segments of DNA in only hours and is automated through a thermal cycler. This can save time and allow analysis to be performed. PCR has many real-life applications, such as in forensics, clinical diagnostics, and agriculture. PCR can be used to detect mutations in humans or home pets. This can show that you have a disease before you even develop symptoms. Or maybe if you don't have any symptoms at all. Some of the analysis can be performed in a small lab. However, reversing the mutations, which is becoming easier because of the technology called CRISPR-Cas9, cannot be performed in a small lab, and it is not widely available to the public either. Special thank you to Avery Holmes, a chemistry teacher in the United Kingdom, and to Daniel Freed, who graduated from Yale University and founder of the project Biochemistry Literacy for Kids. Thank you. Money for the seminar went to Oakland Zoo to feed animals. Bye.